Thank you, Beth Ann. I also am afraid of you, and I think that what I've decided to do is to try to summarize some of the key bullet points or take-home messages from the talk at the very beginning, because if I get tackled off the stage or hauled off or something, I won't be able to finish. So I'll just do that at the beginning. So I think some of the key points I'd like to get across this morning are that there is enormous evidence to say that autism etiology has a really substantial genetic component. It's clearly not all genetic, and that's in part the basis for this symposium today. But genetics plays a key role in it. The genetics is enormously complicated. Everything about autism seems to be complicated. The clinical presentation in autism varies enormously, as most of us know, and the genetics is turning out to be as incredibly complicated as the phenotype is. And we think that the genetics, the underlying genetic architecture of autism, is going to be connected to that difference in how the clinical presentation, what the difference in the phenotype is. So that's another thing. The third thing is that, and more fleshing out this last point in terms of the complexity of the genetics, is that as we're identifying some of the different categories of genetic findings, and we're sort of seeing how these things integrate together to provide more of a comprehensive picture of what's going on, I think that we are at a point now where there is really incredible amounts of progress being made. It's been a very frustrating last, say, 5 years or so when progress has been very slow, but things are really picking up, and there's a lot of things coming out. So having said that, I'll just point to some of the early data that says that autism is, at some level, a genetic disorder. And so this comes from twin studies back in the 70s, beginning back in the 70s, that compared the concordance of autism in identical twins or monozygotic twins versus dizygotic or fraternal twins. And basically, depending on how one defines whether a child is affected with autism, how one defines autism in this case, that identical twin pairs were concordant 60% of the time, that is both affected with autism, and up to 90% of the time if the definition of what autism was or being affected is loosened significantly. But with fraternal twins, it's a much, much lower number. In fact, in some studies, it's even been 0%, but more along the lines of 5 to 10%, so quite a bit less. Now, an important thing in terms of genetics is what the risk of a subsequent child born to a family in which there is a child with autism to be affected with autism. And I think the studies are evolving, and I think the baby SIB studies are going to be incredibly important in this regard, but it seems like the recurrence risk for kids in these families is going to be greater than 10%. And that's really substantial when taken in the context of what we know about the overall population prevalence, which I'm not really getting into. I figure this will be something that Craig Newshafer is likely to address. But with numbers along the lines of 1 in 150 in terms of people being affected with autism for the overall spectrum, or 1 in 500 for narrowly defined autism, which is more typically what recurrence risk ratios are based on, or relative risk ratios are based on, you come up with a relative risk ratio, or lambda S, for sibling relative risk is 50, which in comparison to other say psychiatric and neurodevelopmental disorders is extraordinarily high. So it's really thought to be one of the most genetic of such disorders. So bottom line, it's a very genetic disorder at some level. And so to flesh out some of these ideas of how the genetics are complex can't really be done in 22 minutes, but I can try to touch on some of the issues. So things like Fragile X syndrome are an example of things that are termed phenocopies, but essentially mean related disorders, disorders that have features of autism or that in which individuals present with features that are seen in people with autism. And Fragile X syndrome is one sort of classic example of this kind of thing. So again, there are features of autism, but there's a distinct etiology, a known gene in this case that's involved that isn't really the case for what we would think of as idiopathic autism. Or autism without a known specific cause. An important concept that's been developed over the last several years, particularly by Judy Miles, a clinical geneticist in Missouri, is this notion of essential versus complex autism. And basically, complex autism is a situation in which people that are affected tend to have dysmorphic features. In particular, they're much more likely to have chromosomal abnormalities, other medical comorbidities that are more serious than or more profound than is what is seen on average. So that's an important distinction, I think, as we think about evaluating and diagnosing kids, and certainly for purposes of including them in genetic studies. Locus heterogeneity is a fancy word that means basically there's a lot of different genes involved. Genes probably in virtually every chromosome are going to be involved at some level in autism, be they in rare cases or involved in a much more frequent way or prevalent way in contributing to disease susceptibility. Oligogenic inheritance, another fancy word that basically just means that as we think about for complex diseases, which autism is a complex disease, the idea that a gene that contributes to disease is not causing it in most cases, we presume, but it's somehow conferring additional risk for the disorder. So if there are many genes that have this property and that contribute to autism susceptibility, oligogenic inheritance is a situation in which different families may possess different sets of these genes that are some number of which may be passed on to their kids that may then play a role in risk. But it's going to be different sets of genes in different families for that kind of situation. Another thing that's important is we're beginning to understand much more now is the fact that people that have studied complex diseases for a long time have thought predominantly along the lines that common versions or common forms of a gene, which are termed alleles. Allele is just an alternative or one of many different forms of a gene. That common versions or forms of a gene, common alleles of genes, are what are most relevant to disease risk. In fact, that's likely to be the case in some situations, but it's clearly not going to be the case in others. And so we have to think about, as we pursue trying to find these genes, in which instances are common variation at a set of genes important versus where does rare variation play a role in this. And I'll try to elaborate on that a little bit. There's possibilities for gene-gene interaction, which just simply means that the effect of one gene on disease susceptibility is dependent upon something that's happening at another gene or genes elsewhere in the genome. And gene-environment interaction is certainly going to be very important. I think the problem at this point, notwithstanding what Pat Levitt is going to tell us in the next talk, is that until we really have a handle on what some of the genes are, it's going to be difficult to make firm conclusions about gene-environment interactions. So, as I indicated earlier, people that have been searching for genes in complex disease, as well as autism, for a long time have been thinking along the lines of common alleles or common forms of a gene that may be involved in disease risk. So what have people been doing to try to address this problem? One of the earliest and areas that received a lot of attention from genetic researchers was to do so-called genome-wide linkage screens, which is basically a way to try to identify regions of chromosomes that are believed to harbor some kind of a genetic risk factor. So basically you take families with 2 or more affected individuals. In autism, typically it turns out to be siblings, but it can be any any relative scenario, can be cousins, for example, but families with multiple affected individuals, and you scan their genomes with DNA markers that allows you to track inheritance of all of the chromosomes throughout a family, but then spread out over a large collection of families. And basically, you're trying to identify regions of chromosomes that are sharing a common inheritance in a family, but again, this phenomenon across this entire set of families that you're working with. And once you've identified that region, then in theory, it's easy to go in and find the gene, the allele, if it's one allele, that is what's causing that effect. In reality, it's not as simple as that, and it's quite challenging, in fact. What's a major activity going on now, again, that's very much predicated on the notion of a common allele at a gene that's important in terms of risk, is genome-wide association studies. And much in the way that genomes are scanned with genetic markers for doing linkage studies, in doing association studies for various reasons, one has to use much larger numbers of markers. So more than 1 million DNA markers, for example, are used to try to identify these things. And basically, you're trying to find instances in which there is a form of the gene, an allele of a gene or genes that is seen more frequently in individuals with autism than they are in controls, or if you're using families to do this, ask this question, that are inherited more frequently by people with autism than you would expect by chance. This slide is a figure that came out of a recent review by Dan Geschwind and a fellow in his lab that makes the point of, these are all the chromosomes, the different chromosomes in the genome, and you see little red or green or yellow marks where next to regions that have been implicated based on one kind of study or another. And this maybe is not the best example of such a figure, but it makes the point that there are a lot of different regions across the genome that have been implicated for one reason or another. I think two you will hear about today are chromosome 7, which harbors, among many other, the gene for the MET receptor tyrosine kinase, which is Pat Levitt's group has been doing a lot of work on, and chromosome 17, which has been implicated by linkage and other findings. So again, common disease, common allele. Other kinds of studies that are pursued are candidate gene studies, in which people, acting on the basis of what is known about what's different in autism, so something about neuropathology, so anything that's known that can point you to a function or a pathway, something that might be dysregulated or dysfunctional in the disease, that might provide a basis for a hypothesis, for targeting a specific gene, then you can go to that gene and ask the question of whether that gene has variants. Does it have forms, alleles that are present in cases but not in controls? Are there mutations that are unique to autism, or are there alleles that are present at higher frequencies, and so forth? And a couple of examples of such pathways are those involving neurotransmitters for serotonin and gamma-aminobutyric acid, or GABA, and certainly there's some neurobiological developmental correlates that relate to the MET gene that I'm hoping Pat will talk about. And so again, you can then test those individual genes for involvement. But as over the last few years, there have been a series of studies involving different genes that have really opened the door to the idea that common variation is not going to be the answer to everything. And it's a complex picture. So the flip side to common disease, common allele hypothesis is common disease, rare variant hypothesis. So there's no law that says that there has to be a common variation at a gene that influences disease risk. And so it's also quite possible, as has been the case, shown to be the case in many single gene disorders, that there can be rare variations, rare mutations, for example, at that affected gene, that affected protein that can cause disease or confer risk for a disease. So some of these have come out through, some of these observations have been made through candidate gene studies in which people have been screening those genes that they think to be important in pathways that they think to be important for evidence of disease-related variation, and have found such things. Other rare variant kinds of things, people for many years now have been using observations of rare cases, rare patients in which there is some kind of a chromosomal abnormality that might be detected, for example, in looking at chromosomes under a microscope, in which there are translocations or other kinds of defects that can be seen at a chromosomal level. And by trying to identify the genes around where that chromosomal breakpoint is, the hypothesis goes that you may find a gene that has a broader relevance, not just in this one case, but in general. And that hasn't been terribly helpful, but I think it has borne some fruit. Something that has turned out to be of enormous importance and that has just evolved in the last couple of years is the idea that a class of variation called copy number variation, or CNV, is enormously important in terms of a class of genetic or genomic risk factors. And copy number variation is simply defined as a gain or loss of DNA that's 1 kilobase or 1,000 base pairs of DNA or greater. And so that covers, obviously a wide range of things. So it can be 1 kilobase, or it can be many millions of base pairs. And the more DNA that is affected, then in general, the more genes that will be affected, and you have gains or losses of copies of genes. So we're all walking around with 2 copies of all of our non-sex chromosomes, our autosomes, and if you have a gain or a loss in those genes, then that may be a problem. In fact, we're all walking around with CNV, with copy number variation, both duplications or deletions, gains or losses of DNA, and they have no, and most of them probably have no phenotypic or clinical correlation, but some of them do. And so I think as we're finding in autism that this is a major category of risk factor. And so we're really at a point now where a lot of this is coming out very quickly, and we're learning a lot, and there's several papers will be coming out in the not too distant future on this. And I think one of the things that we're learning, or we're trying to do as we try to look for these genes, is we're trying to put information together from multiple different areas. What we know from linkage or association, and where we see copy number variation that is autism specific or autism associated. There may be genes in those regions that have a broader relevance than just those cases that have a copy number variation. There may be mutations, discrete point mutations, or there may be associated alleles, common alleles, that are affecting genes in regions that are affected by copy number variation. So these are all kinds of things that we're looking at. So to try to go through a couple of examples of the use of these different approaches to try to identify genes, linkage, association, common variation, rare variation, copy number variation, and so forth. I want to show you this figure of chromosome 17, and this thing is a linkage plot. So the y-axis, the vertical axis, is basically corresponds to Lodge scores, which is just simply a measure of evidence, genetic evidence, that there is some kind of a genetic effect at some point along the chromosome. And along the chromosome is on the x-axis, or the horizontal axis. And so where you see that great big peak, or series of peaks, blue and yellow, that identifies a point, a position, a genetic position on the chromosome at which there is some kind of a genetic effect. So the point of this slide, or one of the points of this slide, is that this effect is more predominant when analyzing families that contain only affected males, as opposed to, as compared to the entire set of families used in this study, and then as compared to the subset of families that have only, or that have one or more affected females. So we know that males are more frequently affected than females, and so the majority of families are going to have more males than females and so forth. So that's the predominant set of families, but clearly this seems to be a male-biased effect. And there are multiple groups have reported this phenomenon of linkage in this region, and the AGREE group, led by Dan Geschwind, as well as our group, have reported on this region. So the thing that's relevant, I'm getting to the point that's going to be relevant, is that there is a particular gene at the top of that linkage, corresponding to where that linkage peak is, that is important as a candidate gene, as a gene that we think makes sense based on the neurobiology and what's known about altered phenomena that are present in people with autism. So SLC6A4 is a gene name for a protein called the serotonin transporter, abbreviated as SIRT. So why do we care about SIRT? So it's been known since the early 60s, in fact, that about 30% of people with autism have higher levels of the neurotransmitter chemical serotonin in their blood. And so normally we think of neurotransmitters as functioning in the brain, and obviously they do, but serotonin is also found in the blood. It's localized in the platelets, and that's where the serotonin is seen. But in people with autism, there's a significant number of them, 30 to 35%, that have much, much higher levels of serotonin in their blood. Why? And why we think relates to the serotonin transporter in part. There's a genetic aspect of this in the sense that family members, parents, brothers, sisters that are not affected with autism also show this phenomenon can show this phenomenon of so-called hyperserotonemia. And so this sort of says it's a heritable thing, but it's really enriched in autism and so forth. So this is just, it's been a biomarker that people have been following for a long time. There are drugs called selective serotonin reuptake inhibitors, or SSRIs, frequently thought of as antidepressant medications like Prozac, but are used for various disorders that can be useful in treating some cases of autism where there are problem behaviors that, particularly anxiety-related behaviors that are interfering to the degree that requires a treatment. But those drugs target specifically the serotonin transporter, so that's another piece of evidence suggesting that that particular molecule is important. And regarding serotonin in general, the molecule that precedes, that is required for the synthesis of serotonin, tryptophan, when you reduce levels of that in people with autism, based on 1 or 2 studies, that results in a worsening of symptoms in people. So that says that in general, serotonin is important. So this cartoon shows the serotonin transporter. On the left, you see a normal situation, and basically what you're looking at is the terminus of a neuron making a so-called synapse, with the upper part being the presynaptic side and the lower part the postsynaptic, and serotonin, these little spheres, are being released from the terminal there into the synapse. The function of the serotonin transporter is to vacuum up that excess serotonin, or the serotonin that's in the space between the presynaptic side and the postsynaptic side, to vacuum it up back into the presynaptic side. What SSRIs, or chemicals like Prozac, do is to inhibit that activity, to increase the amount of serotonin that's in the synaptic cleft area. So there would be more binding to receptors and more action downstream for that. And so working this back to this phenomenon of elevated blood serotonin, these things are tying together. Serotonin in the blood, the SIRT protein is in platelets, and so it's, we think, related to the phenomenon of higher levels of serotonin in the blood and so forth. So anyway, SIRT and serotonin are important. So to study this protein, basically what we did is to set aside the common allele hypothesis and ask whether there were rare variants at this locus, which nobody had really looked at before in autism. And to try to make a long story short, we found 4 coding variants, or changes in amino acid coding in the protein, and a couple of these, one of these had been seen before. Another one, interestingly, the isoleucine 425 was a residue that had been seen changed in a handful of pedigrees that were segregating, among other things, obsessive compulsive disorder and Asperger's syndrome. So the bottom line is that we did identify rare coding variants that it's difficult to say that they were causal of disease. There certainly were risk variants in this case. They are functional. There's a long set of evidence that points to their functionality and work that all of this work has been collaborative with Randy Blakely's lab. But anyway, so this is an example in which rare variation at a gene is clearly a factor in disease, but it's rare, so it's affecting a relatively small number of people. So there's a lot more work to do with that. So another example of rare variation has been a story that has emerged that implicates glutamate synapses in the brain, or proteins that function at glutamate synapses in the brain. And so basically over the last several years, people have found rare mutations in a subset of, or a handful of genes that function to encode proteins that work at these glutamatergic synapses in the brain. So another example of rare variation. One of these genes called nerexin is one in which we have seen copy number variation. So this CNV class of disease variation is one we're seeing here in the lower part of the figure, where there's the gene for this nerexin-1, both alpha and beta isoforms, and where you see the red lines are the regions that have been, or the region that has been deleted in two siblings within a family. And so both siblings were affected, and so it was an inherited thing, but it turns out to be a de novo thing. That is, it appears for the first time in the children, not in the parents. So apparently it was in the germline of either the mother or the father in this case, but neither of them was had this deletion present in their cells. So this is an example of copy number variation. There are two reports that have documented some of this stuff, and many more in the last couple of years. There's a well-known phenomenon of duplication associated with a region on chromosome 15 that is also important, and I need to sort of move on because Beth Ann is about to attack me, I think. At any rate, this is an example of some data that identifies copy number variation in a region. And while it's difficult to explain all of this, basically the lower part of the figure shows an individual in which this analysis of using microarrays, very high density spotting of DNA on a slide, is showing a region in which there is an apparent deletion or a loss of DNA that is reflected in the schematic diagram above. And basically all of the genes that are schematized up there are affected and deleted in this case. And so this has been one of the sort of big things in the autism genetics world, is this finding of 16p deletions and duplications in autism. It's a complicated story, but it's one that has received a lot of attention, and there were a couple papers that came out on that recently. And these, again, it's the same region there, and just showing the genes that are involved. So I think people now are very interested in the idea that one or more of the genes in this region that's seen in this copy number variation, deletion or duplication, that's in not a huge number of people. It may be as much as 1% of people, but some of the genes in these regions may have a broader relevance and have alleles that are either causal or increased risk in other families that don't have copy number variation. I'll end there just by pointing out some of the people that I've worked with on some of this data and some of this work in my own lab and Randy Blakely's lab. Also collaborate with Pat Levitt and Ed Cook at the University of Chicago, and formerly with Susan Folstein at Hopkins. Thank you very much.